My name is Lindsay Rogerson, and I currently work as an analytical lab manager in fermentation science. I selected my topic on plant microbial niche, specifically looking at fermented olives. This paper, titled Microbial and Biochemical Profile of Different Types of Greek Table Olives, is particularly interesting to me because of a global resurgence of probiotic foods and their effect on the gut-brain barrier. A lifestyle diet known as the Mediterranean diet has been gaining in popularity due to the abundance of probiotic-rich foods, of which table olives are one. The Mediterranean diet also has foods that are capable of improving health. I will start with a brief introduction. The bottom line up front. The purpose of this paper was to establish a microbial profile of different fermented olive cultivars and determine if biochemical properties had any effect on shelf life stability. If established, this would be first known study to combine next-generation sequencing and biochemical analysis on fermented products. The authors approached this by sequencing both bacterial and fungal profiles using 16S rRNA and 18S rRNA sequencing methods. They also analyzed different biochemical properties. The authors were able to establish the microbial profiles of 5 different cultivars using different fermentation and packaging styles. They determined that salt drying fermentation had a microbial profile significantly different than both Greek and Spanish style fermentations. They performed biochemical analysis, which they determined was able to prove that each production line and packaging type affected the final product. Greece is the second country behind Spain in the production of olives and uses about 80% of their cultivated land for its growth and processing. They produced around 230,000 tons of olives in the year 2020. They use these in a variety of different products ranging from fermented table olives and olive oil to cosmetics and nutraceuticals. These are exported around the world and make a large portion of the Greek economy. There are more than 50 known Greek olive cultivars, and only about half are actually cultivated for use. As stated before, the Mediterranean diet in Greece and surrounding Mediterranean countries has an abundance of probiotic-rich foods that are gaining traction as healers of the gut-to-brain barrier. These probiotic-rich foods can improve health issues and diseases such as by slowing digestion, preventing type 2 diabetes, and heart disease. This paper selected 5 common cultivars for their study. Conservalia amphices, which is a derivative of the Conservalia cultivar, Throba Thesau, Halkidiki green and black, and Calmone, or Kalamata as we call it here in the US. The fermentation process allows these cultivars to result in different products. Greek-style fermentation, also known as natural fermentation, is predominantly used. This is done by brining olives for 8 to 12 months or by packing them in coarse salt. A brine is made of highly concentrated salt water of about 10% salt or less. Brining creates an unbalanced environment and triggers osmosis to occur. The high salt concentration will weaken the olive membrane and cause the olives to release moisture. The barrier weakness allows the naturally present microorganisms to extract and enzymatically degrade oleuropein, which is the cause for olive bitterness. Microorganisms such as lactic acid bacteria and yeast will use the nutrients released by the olive as food and release lactic acid and CO2 as a byproduct. This will lower the pH. The lowering of pH is desirable in fermentation because it will create an uninhabitable environment for most foliage and pathogenic microorganisms. Other fermentation processes performed on the olives used in this study are done differently than the Greek style. However, the result will be the same. In Spanish style, olives are placed in an alkaline treatment such as lye or by being placed in a sodium or potassium hydroxide solution to remove the bitterness. The olives are washed and rinsed prior to brining. In California style, olives are stored in brine and then are subjected to lye treatment. This lye treatment will darken them because of oxidation. The olives will then be washed and color fixed prior to brining. If you've ever had an olive that's been canned and compared it to any of the olives that you find in your cheese or salad aisle, the taste that you differentiate between the two is coming from this process of lye oxidation treatment. Fermentation is a natural process which is begun by the microbiota present on the fruit, or in this case, olive, and in the environment. Confounding factors such as salt concentration, pH of the brine, temperature, storage conditions, and more can play a role in the fermentation capabilities of microbial population. Methodology. Table 1 shows a breakdown of sample cultivars, regions, color, fermentation type, and storage type. 18 table olive samples were selected from 5 different cultivars: Halkidiki, Thuributhaso, Calimone, Emphasis, and Conservolia. 2 different fermentation styles were selected for Halkidiki cultivar. These samples were either packed or unpacked in a variety of different storage conditions. To extract the DNA, 20 grams of each olive sample was homogenized to a pulp in double distilled water using Polytron homogenizer. Microbial DNA was extracted using 400 microliters of the homogenized pellet with the Zymobionics DNA mini prep kit. Zymobionics DNA mini prep kit is immediately ready for microbiome or metagenomic analysis. It achieves lysis by bead beating. We learned in Lecture 3B, Module 3, that the benefit of bead beating is to ensure unbiased lysis. We also learned that chemical enzymatic lysis can lead to unrealistic representation of organisms in later metagenomic analysis that is not reflective of actual abundance. Cubit 4 fluorometer was used to quantify the microbial DNA concentration and assess quality. The kit selected for use was Cubit double-stranded DNA broad range assay kit. This kit is used for broad-range double-stranded DNA with a quantification range of 4 to 2,000 nanograms. It is highly selective and can be used for samples with low concentrations of double-stranded DNA and any common contaminants. Two amplicon metabarcoding libraries were built according to a method developed by Michaelito et al. Bacterial identification was determined by sequencing the V3-V4 hypervariable regions of the 16S rRNA gene. Fungal identification was determined by sequencing the V7 to V8 hypervariable regions of the 18S rRNA gene. Remember from class that there are 9 variable regions, and these regions are standard for identifying taxonomic units by next-generation sequencing. The primers that were selected were based on previous NGS diversity studies. The V3 region was selected for bacteria using the primers dBact0341BS17 and dBact0008AS16 because they are capable of amplifying bacterial, or what we call prokaryotic, genes. The V7-V8 region was selected for fungi. They used universal primers because they wanted to make sure they could cover the broadest spectrum of the eukaryotes while also completely excluding the prokaryotes. Sequencing was done using Illumina standard protocol for 16S rRNA, and a secondary analysis was performed using MeSeq platform for paired end sequencing. The use of MeSeq for secondary analysis works to overlap the ends of each read to generate high-quality full-length reads. Bioinformatics and data analysis was performed using KIME2 software. We started with multiplexed paired end reads. The paired end reads, barcode, and sequence were uploaded to the software. Adapt was then used to remove adapter sequences by trimming. The adapter sequences are indicative of NGS alignment errors and low quality reads. The quality paired end sequences will then be used in Data2 algorithm, which removes PCR primers, dereplicates, and denoises filtered reads. This will be put into a featured data table, which have been placed into the frequency table, can be used to look at diversity in 2 different ways. Alpha diversity will be used to determine diversity within a sample, while beta diversity will determine diversity between different samples. Alpha diversity will look at metrics such as the distribution of different organisms shown by richness and also how they are distributed in terms of abundance shown by evenness. Beta diversity will look at the distance or dissimilarities of samples. This paper used ordination to create principal coordinate analysis to group samples with similar microbial profiles. Already demultiplexed and dereplicated our joined pairs. The next step is to remove our chimeric sequences or artifact sequences formed by two or more biological sequences incorrectly joined together. These paired sequences will then be merged, resulting in the final ASVs, or what we call amplicon sequence variants. ASVs are the same as operational taxonomic units, or OTUs. ASVs were then aligned to Silva 138 database with 99% similarity. Any remaining unnecessary/unassigned sequences were removed from the ASV table. This resulted in a .biome file that was then capable of uploading to R version 4.2.1 for phylogenetic analysis. First, the import biome file was uploaded into R version 4.2.1 for phylogenetic analysis. The raw data was then sent into pre-processing, and then from the processed data went directly into direct plots for plot richness and plot bar. Both of these are alpha diversity indice markers. Then, ordinate was used with phyloseq data to create principal coordinate analysis, or PCOA, by applying the Bray-Curtis distance method. This determines whether samples are similar to similar and semi-similar. Plot ordination is considered beta diversity. Touch on the biochemical analysis as it is important to the presence of or absence of specific microorganisms. However, since it's not directly related to the focus of this microbiome course, I'm not going to really go into the method specifics. However, feel free to ask me during our discussion. This is something that I do all the time. pH during the fermentation process is important because lab bacteria and yeast will produce acids that will then lower the pH. This is important for the safety and quality of the final product, and it also discourages the growth of spoilage and pathogenic microorganisms, and thus extends the shelf life of the product. Total polyphenolic content is important because polyphenols will produce many of the antioxidants and also indicate antimicrobial activity that helps to prevent unwanted pathogens and spoilage microorganisms. The antioxidant activity will display the probiotic potential of the final product, and it's also important to both the consumers and producers. The quantification of these individual polyphenols is very important because some of them are known for their bitterness of the fruit, and they will note when it is degraded during the fermentation process. And they can also be important to the biological properties that are linked to many health benefits. A high concentration of polyphenols indicate low levels of lab bacteria and dominating yeast growth. I would like to head into the results. 2 shows the number of ASVs and alpha diversity indices for each sample based on 16S rRNA amplicon sequencing. Raw reads were obtained from 16S rRNA about 897,320, which were then reduced using KIME2 to an average 20,650 quality filtered reads per sample for bacterial identification. Microbial diversity was measured using alpha diversity indices for abundance and richness. Some background on the alpha indices in this table. The first column holds our sample names. The second column holds our observed ASVs, or number of observed amplicon sequence variants, which are alternatively called OTUs. The third column holds the Shannon value, which is the estimate of microbial species diversity within a sample. A higher Shannon value signifies a greater level of community diversity. The fourth column holds the Simpson value, which measures the concentration or richness of a species within a community. The closer the value is to 0, the least diverse it is. The closer the value is to 1, the more infinitely diverse it is. The fifth column holds the inverse Simpson value, which is a quantitative measure that reflects how many different species there are in a community. The higher the value of D, the greater the diversity. Calamone 3 and Amphisis 2 had the highest observed ASVs indicated by the red circles in column 2. They also had the highest Shannon value index shown by the red circles in column 3. These indicate high species richness in these two samples. Calamone 1, Calamone 2, and Halkidiki 1 all had the lowest number of observed ASVs. This is indicated by the blue circle in column 2. Calamone 1, Halkidiki 2, Thesso 1, and Conservolia 3 all held the lowest Shannon index values as shown by the blue circles in column 3. This suggests that they had low diversity. Amphisis 2, Calamone 3, and Halkidiki 6 showed higher bacterial diversity, and this is also shown by the Simpson index circled in red and also shown by the inverse Simpson also circled in red. Remember, the closer to 1, the more diverse it is. Likewise, the larger the value of the inverse Simpson, the greater the diversity. The authors broke the microbial data from 16S rRNA sequencing into phylums, class, taxonomic order, family, genus, and species. They elected to graph the distribution of microbial families present in each sample against the normalized relative abundance percentages. Remember that fermented foods are typically lactobacilli dominant due to the natural presence on food and in the environment. They have an ability to thrive in low pH environments. Of the families found among the samples, Cons 1, Cal 1, and Amp 3 were the richest in lactobacilli at above 96%. The authors stated that Cal 3 was also rich in lactobacilli above 96%, but if you look at the figure 1, you will see that lactobacilli is significantly less than 96% abundance in that sample. However, next to it, we have Cal 2, which does have a presence of lactobacilli above 96%. I believe this is what they meant to group with Cons 1, Cal 1, and Amp 3 in this example. Conservolia samples were, on average, the richest in lactobacilli family as well. Thesso and Halkidiki were the only cultivars not dominated by lactobacilli. Instead, Leuconostoca C was found to be the most abundant in Halkidiki and the second most abundant in Thesso. Each Thesso sample had their own number 1 family, and this was not the same across all samples. Other abundant families were Enterobacteriaceae, Yersinia and Erwinia. To note that here the authored results are not clear as the bar plot shows contradictions. For example, there is a large presence of Leuconostoxae in Halkidiki samples 1 through 3, but not so in examples 4 through 6. They mentioned prior that Halkidiki was most abundant in Leuconostoxae, but if you look at the bar plot, this is a misleading statement. Likewise, the author stated that Thesso was one of the only cultivars not dominated by lactobacilli. However, if you examine the bar plot again, you will see that Thas 1 is the most abundant in lactobacilli. The authors noted that 2 samples, Halk 6 and Cal 3, shared similarities in their packaging and storing. They also shared bacteria from the Commonodaceae family, which were not detected in other samples. However, if we look again at the bar graph, we can see that this bacterial family was also detected in AMP2 and in Cons 3. To me, this showed the authors really wanted to use this as proof of packaging and storing having an effect on microbial profile. Both AMP2 and Cons 3 samples, which are stored and packed differently than Halk 6 and Cal 3, contradict that argument. Figure 2 shows principal coordinates analysis, or PCOA, for the 16S rRNA amplicon metabarcoding data for all samples. All Thesso samples were clustered near Halk 2. You can see this circled here in green. This indicates that they shared bacterial similarity. Halk 1 and 3 were proximal to this cluster, as shown circled here in blue, which shows that they might have some similarities in bacterial profiles to the first green circle. However, HALK6 and Cal3 formed a cluster away from all other samples. This is shown here in red. This indicates that their bacterial profile is significantly different from the others and are more similar to each other's. There is no other clear grouping, even though HALK5 and Cal2 circled in purple were in close proximity. Focuses on the number of ASVs and alpha diversity indices for each sample based on the 18S rRNA amplicon sequencing. Remember that the 18S is for the fungal diversity. Raw reads were obtained from the samples for 18S rRNA, about 1,221,185, which were reduced using KIME2 to an average 29,161 quality filtered reads per sample for fungal identification. Fungal diversity was measured using alpha diversity indices, just like bacterial diversity. Cal3 had the highest observed ASVs, followed by HALK6, as shown in the second column by the red circles. CONS1 had the lowest, as shown by the blue circle. Sample Cal3 and HALK samples 3, 5, and 6 had the highest Shannon and the highest Simpson indices, as shown by the red circles. This indicates high species richness and evenness. The authors did not discuss the lowest indices for 18S rRNA. This is something that I could not determine as to why. The authors deviated from creating a bar plot using family relative abundance percentage and instead used genus for 18S rRNA. They did not provide a reason for the change, but I assume this is due to having too many genus or species for bacteria to make the bar plot readable. 18S rRNA does not have nearly as much library buildup from research as 16S rRNA does, which could be a reason for why it was reasonable to plot at the genus level. Furthermore, the author stated that samples of the same cultivar showed great biodiversity at the genus level. To begin with, Meniscus, which is a mold, was the most abundant genus in AMP1 at 98.7%. Starmella candide clade was predominant in AMP2, and all HALK6 samples had great biodiversity. Cal1 and Cal3 were dominated by Saccharomyces, which is a yeast, and Cal2 and AMP3 were dominated by Pichia. The authors then discussed variability at the species level, but they did not offer this bar plot in either the paper or in the supplementals. They noted that the second most popular fungal genus, Malassezia, was only found in CONS1, but like the bar plot in 16S rRNA, other samples did actually display Malassezia. CONS1 shown here, you can see the Malassezia is the green at the top, but this is also the same green seen in the Cal3 and also in HALK6. Figure 4 shows principal coordinates analysis, or PCOA, for the 18S rRNA amplicon metabarcoding data for all samples. At the top in green, we see Cal3 and HALK6 clustered together. This is indicative of similar fungal profiles. All Thasau samples, along with HALK1 and 3 samples, were closer together than the rest of the samples. This shows that they have a similar fungal profile. HALK2 was still a proximity to them, but it was still different enough that it's not going to be clustered together with them. There were no other clear clusters other than the AMP2 and HALK5, circled here in purple. The bar graph of Figure 5 shows the biochemical values of the samples expressed as an average for each of the cultivars. The authors further took the 5 cultivars and broke the Halkidiki into 2 because it had 2 different fermentation styles. This can be seen circled in yellow, and this stands for Halkidiki Thruba and Halkidiki Green. So that's for your black and your green olives. Thurba Faso and Halkidiki Thurba had significantly higher pHs, which is noted by the A letter. And you can see this in the blue and in the silver color. Halk 6 and Cal 3 had the highest total phenols. However, figure 5 has averaged out the cultivars, so it's not verifiable using this figure. Instead, the authors posted the results from each sample prior to averaging the cultivars. Table 4 shows the biochemical values of the 18 samples analyzed. The results are expressed as a mean plus or minus the standard deviation. All biochemical analysis were performed in triplicate. 6 and Cal 3, shown by the red rectangles in column 2, had the highest total polyphenols, while Cal 1 and Thas 2, shown by the blue rectangles in column 2, had the lowest. Antioxidant activity was in accordance with total polyphenol content, as it showed a reverse pattern with Cal 1 having the highest, displayed by the red rectangle in column 3, and Cal 3 with the lowest, as displayed by the blue rectangle in column 3. 3-hydroxytyrosol, alurapine, alucanthal, and oleacine were interpreted from each sample's HPLC data. The highest amount of hydroxytyrosol was found in Cal 3, Halk 6, and Halk 4, as can be seen in column 4. The lowest was found in Thas 1 and 2. Oleacine was highest in Halk 2 and Thas 2. And this is interesting because both samples used the salt drying fermentation method. Alurapine was mostly undetected in samples, and this makes sense because if you remember, fermentation is done to remove the bittered form of the olives, and this is a bittering compound. The highest was found in Cal 2 and Thas 3. The highest alocanthal levels were performed in 2 Halk samples and in Thasius sample. Discussion. For the bottom line up front, this study aimed to combine next-generation sequencing of microbial communities found in fermented Greek table olives and biochemical analysis to determine if packaging and storage types had any effect on the final product. The authors were able to match the variability of the microbiota in each cultivar and fermentation style with previously published research studies. The study also hoped to determine that packaging and storage conditions had an effect on the final product. However, I found a better argument could have been made for the fermentation style's effect on the microbial community in the final product. An example for this is that Amphisis packed by the same processor in all 3 samples, 2 of them had a spoilage microorganism. This is something that could display that the microbiome is affected by the processing environment as well. This is a variable that was not factored into this study. Another limitation that the study had is that there is a large variability of the species among Spanish and Greek style fermentations. This made it impossible to determine whether those methods had any effect on each cultivar. This indicates a need to reduce other confounding variables in future sequencing. Many of the confounding factors showed an effect on the phenolic content, including the cultivar, degree of ripening, and the method of fermentation. Calamon had the highest total phenols within the same fermentation style, while Amphisis had the lowest. This indicates that phenolic content was more affected by the cultivar than by the fermentation method. Furthermore, there was no significant difference in antioxidant activity between the samples. The authors were not able to conclude if this was due to the cultivar or to the fermentation style. This could be an area for future research. Supplements show more of the arguments than what is shown in the paper. I would have added the average of the cultivar bar plot as well as the HPLC chromatogram as part of the supplemental section, and instead included the 2 PCOAs of 16S and 18S rRNAs for the fermentation type, as I believe that this would back up their results and discussion in a way that would show the microbiome of the fermented products and their biochemical properties are related to shelf stability. Fermentation styles are known to impact shelf stability, and this was observed by this study when they drew the two main conclusions. First, that salt-dried fermentation style had a microbial profile significantly different than both Greek and Spanish fermentation styles. And two, the biochemical analysis showed that the production process, otherwise known as a fermentation style, and the type of packaging affected the microbiome, and thus the stability of the final product. I do not believe that they made a strong enough argument for a shelf study or packaging. In my opinion, the authors lost focus of their purpose and could have made a better argument had they not mishandled their data and actually approached it with the fermentation process as one of the most important factors. An additional factor could be a true shelf life study with time as a variable. So I see more data backing up the fermentation style affecting the final product than the packaging type based on their data. A shelf life study using time as a variable would be very beneficial to strengthening this paper. The authors did note that Macalado et al. Performed a shelf life study on Greek-style table olives that had been stored under a modified atmosphere. And this is something that they didn't actually use in their discussion, but it should have been there. Despite this, the study was significant in combining microbiome sequencing and biochemical analysis towards fermented foods. They were able to show that the fermentation style affected the microbiome of the final product, which contributes to the biochemical aspects of table olives. They need to add the factor of processing environment as a variable. They also need to revisit this data as more genomes are sequenced, and additional biochemical tests such as moisture content, brine salinity, etc., could be added to make this more robust. The study was significant in combining microbiome sequencing for bacterial and fungal communities present in fermented Greek table olives and biochemical analysis. The authors failed in their second objective, which was to determine that packaging and storage type affects the final product. Overall, how the authors approached their experimental design was lacking the key confounding variable of time in a shelf life study to prove that packaging type and storage affects the final product microbiome. Some questions that I had to get our discussion going. What other confounding variables could the authors have integrated into their biochemical analysis? They could be either biological or technical. One of the things I considered was time as a variable or the processing environment. Second, what might be the benefit of selecting a different assay rather than qubit double-stranded DNA broad-range DNA assay kit? Qubit offers an extended kit that has a wider range. My third question, what limitations occur when specifying primers for specific variable regions? The bacterial genome was selected for using specific primers, while the fungal genome used universal primers. My fourth question, why do you think some cultivars had high species richness and evenness over others? The 18S rRNA results didn't focus on cultivars lacking richness and evenness, and it actually left them out entirely. My fifth question, and the most important question from this entire paper, results from the sequencing data did not seem to be appropriately interpreted by the authors. Did you find that the authors mishandled their data? Thank you so much for joining me in this paper discussion, and I hope to converse with you more in the coming weeks. Here are my references. Page 1 and page 2. Thank you so much for your time.